Specification point 1.7. Explain the mechanism of enzyme action, including the active site and enzyme specificity. To successfully answer any GCSE question linked to this specification point, you will need to know what an enzyme is and how it works. Enzymes are biological catalysts. A catalyst speeds up a reaction without becoming part of a reaction. You will learn more about this in chemistry. Each enzyme is specific to its substrate. This means it will only work with one substrate. The enzyme is a protein with a specific shape and active site. The substrate binds to the active site. The enzyme catalyzes the reaction. The products leave the active site while the enzyme remains unchanged. The enzyme can then continue to bind to another substrate. As already mentioned, enzymes are specific to one substrate. This is known as the lock and key model. It is known as this because it explains how a specific substrate will fit the active site of an enzyme like a key fitting a lock. Only one specific key and identical copies can open your front door. Similarly, only one substrate can fit into the active site. Explain why no product will form if the enzyme is mixed with substrate Q. If we combine what we have learned over the last couple of minutes, we can easily answer this question. Here we can see two substrates and an enzyme. This circle-shaped substrate will not fit into the enzyme's active site. The enzyme is only specific to this substrate. The first thing we learned was that the enzyme's active site is specific to a substrate. This means enzymes will not bind to every substrate. We also learned that if an enzyme-substrate complex is formed, then the enzyme catalyzes and speeds up the breakdown of the substrate. This also means that if the substrate does not fit, no reaction will occur. Let us understand this alongside the mark scheme. In this question, you get first and second mark for stating that active site of the enzyme will not fit substrate Q. In other words, it is not complementary. The third and final mark for this question will be rewarded for stating that the enzyme cannot cause a reaction or catalyze the reaction.